So I am an analytical chemist working as a senior researcher at the Willox Lab. We are a small molecule LC-MS analysis laboratory located at the Biomedicum at Karolinska Institutet. We have a combination of both non-targeted and targeted LC-MS platforms working in a biomedical institution. So my role includes supervising but also performing all parts of the analytical workflow, from study design to data collection, analysis of the results and reporting in unscientific discussion with the collaborators. Our main challenge is to keep everything working smooth and being able to always provide the best results.
What would you say is your decision process when deciding to buy a new product?
So we study which products available can cover our needs, and then we think of ways of benchmarking them and how we can incorporate them into our workflows. If it is possible, we test them, we do pilot studies, and if everything works, we incorporate them into our workflows.
What characteristic does a product or a company have for you when you decide to choose it?
So science must be reproducible. So everything that we incorporate into our workflows should be directed to achieve reproducibility. In addition, what we need is that the companies are responsive to our needs, because there are different deadlines that we must achieve. So it is very important that we are sure that everything will be robustly and on time delivered to us. For us, Biotage has a great customer service that has allowed us to quickly solve any problems that we have faced over the years. Biotage is also continuously innovated their products, such as this new TurboVap system, and they are in contact with us, and they always take into account our opinions when developing these products, and that is very important for us because this allows us to get a product that is tailored to what we need.
When deciding to choose Extrahera, what was the problem or the challenge that you need to overcome?
At our laboratory, we have different LC-MS targeted platforms. One of the families of compounds that we analyze are oxylipins, which is a family of lipid mediators involved in many biological processes, such as inflammation. For that compounds in particular, we require sensitivity and specificity because they are present at very low concentrations in the human fluids. So we need to perform solid phase extraction, both to clean the samples and also to pre-concentrate the compounds. We needed a system that allowed us to perform this task in a reproducible way. And we also needed that this system allowed us to perform this task in the required throughput and with the required reproducibility that our projects demand. Extrahera helps us accomplishing our goals because it allows us to free up some time with repetitive and cumbersome tasks that we can employ to perform other tasks at the laboratory. I mean, I think for now, for us, everything is quite good. I mean, I was personally very surprised that everything works so well when moving from the manual extractions, because like these automation processes, you know, the first time they go and they get stuck in the middle, and then so I mean we have had problems, but taking into account all the work that we do, everything has worked pretty well.